Okay, good afternoon. This is Nelly Poyak from Dana-Farber. Welcome to the DFHCC Connect Science seminar series. As a reminder, the seminars are recorded and will be posted on the website that's indicated in this first slides. It usually takes about a day to do this, so you can go back and check. As also a reminder, please post your questions in a Q&A function in Zoom, not in the chat. And the speaker will have access to the questions, and we try to answer as many as we can live, and then the remaining questions he can get back to you after the seminar. So it's my pleasure to introduce my colleague from Dana-Farber, Miles Brown, who's the director of Center for Functional Cancer Epigenetics at Dana-Farber, and also an Emil Frey Professor of Medicine at Harvard Medical School. Miles is also a member of the National Academy of Sciences, National Academy of Medicine, and a fellow of the AACR Academy, and has received numerous awards. He's been a leader of dissecting and understanding the function of hormone receptors in breast and prostate cancers, and developing and utilizing technologies that allow the mapping of the targets of these receptors. And Miles is also, I'm happy to have him as my lab neighbor. He's been my personal advisor ever since I studied at Dana-Farber, and really good colleague and friend over the years. So Miles, thanks for joining us today, and we're looking forward to your talk that's entitled Feedback Loops Limiting Effective Breast Cancer Therapy. You're muted. You have to unmute yourself. Yeah.
Okay, you can hear me now?
Yes.
Yes, we can hear you.
Yes.
Thanks, Nelly. So it's a great pleasure to present today some work from my lab, really sort of with a therapeutic theme, trying to understand really what it means for certain pathways to be dependencies in breast cancer in this example, but what understanding the true nature of the pathway can lead to new sort of therapeutic potential insights. So I have these disclosures. I consult for and get sponsored research support from Novartis. I'm on several biotech SABs. So in our lab, we've thought about what drives sensitivity and resistance to therapy in breast cancer, prostate cancer, and now in ovarian and other diseases. But I think these basic principles hold true across cancer types. So the first has to do with lineage-specific differences, and that really came to us by thinking about the dependence of breast cancer, 2/3 of breast cancers on estrogen signaling and estrogen receptor, and the vast majority of prostate cancers on an androgen receptor. And what these dependencies are residual dependencies of the normal tissue of origin. So growth and development of the mammary gland is dependent on the estrogen receptor on estrogen, and likewise in the prostate, it's dependent on the androgens receptor and androgens. So these lineage-specific differences are extremely important and really explain a lot of the different sensitivities across different cancer types. And then perhaps the most common and potentially the most important mechanism of therapeutic resistance to targeted therapies has to do with clonal evolution. And of course, this is not a new concept, but for breast cancer and the estrogen receptor, work from several labs, including Renate Jesselson, when she was in my lab, really demonstrated that mutations in the target are often the first approximation as the most likely mechanism of resistance to a targeted therapy. So her work really identifying the importance of estrogen receptor mutations that render them resistant to estrogen deprivation or to specific ER antagonists, really clinically is turning out to play a very important role in therapeutic resistance in breast cancer. And then an idea that I've been looking for really my whole career, in which people who became interested in so-called cancer stem cells thought was a new idea in the sort of early part of the century, but it's really a very old idea. And that is that there might be hidden under a mass of a tumor a small number of tumor-initiating cells, cancer stem cells, cells that can form a new tumor that may actually be target independent. And surprisingly, in the breast cancer world, work from several groups really focused on chemotherapy resistance and the potential role of cancer stem cells, really for which there's, I would say, no clinical evidence whatsoever. But for hormone independence, I think that's an area where one could imagine a estrogen-independent tumor-initiating cell being important. But the discovery of the estrogen receptor mutations tells us that's not a frequent mechanism, at least in those cases. And then what I'm going to talk about today are really the existence of what I would call hardwired feedback loops. That is, the dependency is tempered by the presence of feedback loops in the signaling pathways of interest. So the first story I'm going to tell you about has to do with a feedback loop involving the estrogen receptor in breast cancer. And this is work from Tengfei Xiao when he was in my lab, published a couple years ago. So in the estrogen receptor world, there's been a lot of interest in the potential role of feedback loops and crosstalk, and that's really partly because this comes from the field of endocrinology, where feedback loops are the norm. Though these are, what people in endocrinology mostly think about are not cellular-based feedback loops, but organismal feedback loops, and that maintains normal homeostasis through feedback between different endocrine organs. In breast cancer, there's been a lot of work over the years identifying potential crosstalk and feedback loops involving the estrogen receptor. And we, a couple years ago, wanted to try to use modern genetic technologies to elucidate what these pathways might actually look like. And to do that, we performed genome-wide CRISPR screens, which I'm sure everyone is now quite familiar with, to identify genes whose loss confers either a negative or positive selection under different culture conditions. And to do this, we took two estrogen receptor positive breast cancer cell lines, T47D and MCF7, and infected them with a genome-wide CRISPR knockout library and cultured them for about 10 population doublings under vehicle conditions, that is hormone-deprived conditions, or in estrogen supplemented conditions, and then compared the frequency of the guides at the end of the experiment compared with the beginning. And when you do that, you first find a large number of essential genes in these two cell lines. Of course, most of these essentials, many of these essentials are so-called pan-essentials, elements of cellular machines that are required in all growing cells, ribosomal subunit proteins, spliceosome, proteasome, et cetera. These are pan-essentials that we found really as sort of positive controls in this type of screen. What was quite gratifying was, in addition to the pan-essentials, we found a number of common essentials that turned out to be genes that we had identified over the year as playing critical roles in the estrogen receptor mediated growth of ER-positive breast cancers, including FOXA1, NCOA3, the ER target MYC, et cetera. If you display this data in a slightly different way, so here the genes are ranked in order of essentiality, and you can see that the breast luminal determining lineage factors, FOXA1, GATA3, SPDF, ER, are all highly uniquely essential that is in a lineage-specific way in ER-positive breast cancer. What was also interesting was looking at the genes whose loss was positively selected. And this allowed us to think about genes that conferred a growth advantage when lost under estrogen-deprived conditions, which can model sort of endocrine therapies such as aromatase inhibitors that reduce serum estrogen levels. And what was again quite gratifying is that genes whose loss and had been previously implicated in endocrine resistance, including tumor suppressors like TSC1 and 2, and F1, P10, were positively selected. What was most interesting for us was the fact that the CSK gene was the most consistently positively selected as being differential under vehicle versus estrogen treated conditions. And we validated that by taking different specific guide RNAs to CSK. So under control conditions, using a control guide of targeting AAVS1 in either T47D or MCF7, under estrogen-deprived conditions, you don't get any growth. If you silence CSK, you get full growth back. If you put CSK back in, you again no longer get growth. So this validated the results of the screens. This also conferred relative resistance to tamoxifen. That is, if you look here, that both in T47D and MCF7 cells, the selective estrogen receptor modulator tamoxifen, which has quite interesting properties in that it only partially inhibits estrogen receptor function because it actually stabilizes the receptor binding to chromatin and allows activation of a subset of its normal targets. But these knockout of CSK rendered these cells completely resistant to tamoxifen. Interestingly, the selective estrogen receptor degrader fulvestrant, which is considered a more complete estrogen receptor antagonist, knockout of CSK only renders the cells partially resistant to fulvestrant. And when we looked at the same cells grown as xenografts, again, you can see that under control conditions on the absence of estrogen, MCF7 cells won't grow as a xenograft in immune compromised mice. If you give them estrogen, they grow quite well. But if you knock out CSK, even in the absence of estrogen, you get quite good growth. Again, supporting the idea that loss of CSK confers estrogen-independent tumor growth. So just to remind you what CSK. So CSK is C-terminal Src kinase. It's an inhibitory kinase of Src family members. So it phosphorylates Src and its related tyrosine kinase family members at a tyrosine 530 in Src, and that inactivates Src signaling. And that's interesting, both because Src is critical for cell proliferation and tumor growth in many different models. It also plays a role in cell motility and invasion. And that was of interest because when we looked at the phenotype of the cells in which we had knocked out CSK, we saw this very striking crescent-shaped phenotype with a large leading edge. And these cells are more motile and invasive than the controls. And this suggests to us that loss of CSK, which was selected for allowing the cells to grow in the absence of estrogen, confer a phenotype that might be associated with increased metastatic potential. And this is an idea I've liked for a while, in that metastasis is not really a selectable phenotype in cancer. But what is selectable is growth and survival. And it turns out that often the genes whose mutation, either loss or gain in case of oncogenes, confer a selective growth advantage, also change the phenotype and can influence metastatic potential. So that's true for this loss of CSK. It turns out it's also true in Renat's work for the gain of function estrogen receptor mutations that confer hormone-independent growth. They also confer a pro-metastatic phenotype as sort of an epiphenomenon. So what was interesting when we tried to study how CSK was regulated is we were able to look at the estrogen receptor systrome from breast cancer cells. So, Shirley, Lou, and I defined the idea of the systrome as the set of targets of a trans-acting factor across the genome that is, in practical terms, the results of an experiment like a CHIP or CHIP-seq, or now a cut and run experiment that is the set of its targets in the genome. And we looked at the estrogen receptor systrome at the CSK gene. We identified a very prominent estrogen receptor binding peak about 10 kilobases upstream of the CSK start site. And that site had characteristics of a transcriptional enhancer because it was enriched for H3K27 acetyl, a histone mark typical of active enhancers, and also was accessible in terms of DNase sensitivity. And when we asked, well, was this enhancer important in playing a role in estrogen-regulated expression of CSK, we did a knockout experiment where we flanked the enhancer with pairs of guides or control guides and asked how did that influence how did that influence CSK expression. So under control conditions, CSK is estrogen induced quite rapidly within 1 hour. If we knock out this enhancer using several different sets of guides that delete the enhancer, it's no longer induced. If you use control guides, it's induced again. So that says that CSK is directly regulated by the estrogen receptor through binding of this ER-bound enhancer about 10 kilobases upstream of its start site. But what was interesting to us was that loss of the CSK enhancer had the same growth phenotype as loss of the CSK coding region. So guides targeting that enhancer also conferred estrogen-independent growth. And again, point to, I think, an emerging, and again, I think from my point of view, something that almost certainly had to be there, which was that cis regulatory mutations turn out to be important drivers in cancer development, and in this case, in therapeutic resistance. What was also gratifying is when we looked at a set of actual human breast cancers that had been profiled by estrogen receptor CHIP-seq for the presence of this estrogen receptor bound enhancer, we found in the vast majority of ER-positive samples, there was strong binding of estrogen receptor to the same binding site. Of course, in a couple of ER-negative controls, there was no binding. And interestingly, the degree of binding of the estrogen receptor to that binding site was different in different patients. So then we next wanted to ask, well, if loss of CSK can confer hormone-independent growth, does that engender any synthetic lethal vulnerabilities? And to do that, we performed a genome-wide synthetic lethal screen where we introduced guides targeting CSK, 2 different guides, or 2 different guides targeting the control site in the absence of Cas9, and then infected the cells with the genome-wide library containing guides for all the genes expressing Cas9, so both genes would be deleted at the same time and cultured under vehicle or estrogen-repleted conditions. And when we do that, we get quite an interesting network of genes that are differentially lost, differentially essential upon CSK loss. And quite interestingly, the most sort of the gene that was most synthetically lethal with CSK was the gene PAC2. Interesting, other genes that have been known to become more essential in the setting of estrogen independence in breast cancer, including ERBB2 and elements of the PI3 kinase pathway, were also synthetically lethal with CSK loss. Interestingly, none of the Sark family kinase members was uniquely synthetically lethal with CSK. And that leads us to the idea that potentially more than one Sark family member is present and there's a functional redundancy. So what's PAC2? So PACs are downstream of CDC42 RAC. They're serine threonine kinases In fact, they've already been implicated as potentially targeting the estrogen receptor. So to confirm that the PAC2 is indeed synthetically lethal with CSK loss, we took, again, individual guides in cells that either had knocked out CSK or not. And then two of the guides here, guides 1 and 3, are good at knocking out PAC2. And those same two guides, in the absence of, so in the presence of CSK, they have really no effect on viability. But in the absence of CSK, we see this synthetic lethality of loss of PAC2. And then we wanted to make sure that the signaling was happening the way we imagined. And to do that, we again compared cells that had either knocked out CSK or CSK had been reintroduced in a rescue construct, and then asked what happened to phosphorylation of both SARC family members and PAC2. So as expected, loss of CSK reduces the level of this C-terminal phosphorylation on SARC family members. That's activates SARCs, and that's indicated by phosphorylation of this tyrosine of 419 in SARC. And also this serine 141 in PAC2 becomes phosphorylated in the absence of CSK. So this led us to some ideas about potential therapeutic combinations. So in this case, we tested both a SARC family kinase inhibitor, saracatinib, that actually has been in clinical development, or a pan-PAC family inhibitor, FRAX 597, which is really a tool compound. And in both cases, loss of CSK sensitized the cells to either the SARC inhibitor or the PAC inhibitor. We also did this in tumor models grown as xenografts. So here in the CSK null tumors, we see some growth inhibition by either the PAC inhibitor or the SARC inhibitor, very modest inhibition by fulvestrant, but quite strong inhibition by the combination of the estrogen receptor degrader fulvestrant and either a PAC or SARC inhibitor. And we were able to confirm this combinatorial effect in a estrogen receptor positive PDX model. So that's the growth under vehicle conditions. The cells are mildly sensitive to fulvestrant. The PAC inhibitor has, again, sort of modest effects, but the combination of fulvestrant and the PAC inhibitor quite significantly inhibits their growth. And we also wanted to ask, was this something we could look, if we looked at clinical samples? So we actually looked at the expression of CSK in primary, instead of primary and tamoxifen resistant samples, and saw that loss of CSK expression is associated with the tamoxifen resistance in clinical samples. And then a gene expression signature based on CSK loss was also a predictor of response to endocrine therapy. So this first story really identifies a feedback loop involving estrogen regulation of estrogen receptor, which directly upregulates CSK. And CSK is a negative regulator of SARC family members. So when you block estrogen receptor, either by removing estrogens with therapies like aromatase inhibitors or drugs like tamoxifen, you actually decrease the levels of CSK, and this leads to SARC family reactivation. And this loop actually goes all the way back up to estrogen receptor. So really what it tells us is that the estrogen dependence of breast cancer cells is modified by this feedback loop. And we can't expect simply inhibiting estrogen receptor to fully inhibit the ultimate growth signals, which in this case involve activation of both cyclin D and MYC and BCL2 as well as key oncogenic targets of the estrogen receptor. And it really says to fully inhibit this pathway, you've got to hit it twice, both potentially at the estrogen receptor and also either at SARC family members or PAC2. So the second story I wanted to share with you is an unpublished work in a different type of breast cancer. So as probably many of you know, breast cancers have been characterized by expression of steroid hormone receptors, estrogen receptor, and progesterone receptor, and whether they harbor the ERBB2 gene amplification. And clinically, that classification is important because patients with those types of breast cancer get different treatments. And a sort of clinically useful but not very biologically satisfying classification of breast cancers that lack estrogen receptor and progesterone receptor and HER2 is that they're triple negative. And again, that's useful mnemonic clinically because those patients won't respond to endocrine therapy or HER2 targeted therapies. But it turns out that that's really a sort of grab bag of lots of different, at least half a dozen different cancer subtypes. So in this 25% of breast cancers that don't express steroid hormone receptors and don't express HER2, it's further broken down to maybe a half a dozen different subtypes based on gene expression and epigenetic state. So finding targets that can be effective in this 1/4 of breast cancers has been challenging, partly because it's very heterogeneous. And I think people should expect to find, if they want to find targeted therapies, they may be lucky to find one that's useful in 5% of breast cancer. So maybe 1/5 of the triple negative subset, simply because it turns out it's quite a heterogeneous group of tumors. Now that shouldn't dissuade people from looking for them because of course we know, for example, lung cancer, those harboring ALK alterations are very sensitive ALK inhibitors, and that treatment is very important, but it's a very small subset of lung cancer. So identifying the correct subset, even though it's relatively small, is still extremely important. And Yuha Lee in my lab has started to look at potential targets in triple negative breast cancer. And what he focused on first was the potential to target FGFRs as a targetable dependency in triple negative breast cancer. And what he found by just looking at publicly available data is that several percent of triple negative breast cancers harbor FGFR amplifications, either FGFR1 or 2, quite commonly sometimes FGF4. They also have been known to harbor EGFR amplification. So for the FGFRs, it may be as high as 10% of triple negative breast cancers. Now, FGFR inhibitors have been studied, at least in early clinical trials in breast cancer, and then results have been disappointing. So while in urothelial cancers and in biliary tree cancers, FGFR inhibitors have gained use and are approved clinically, in breast cancer, most of the data has not really pointed to a very strong clinical effect. So that was surprising to us, given that several breast cancer, triple negative breast cancer cell lines harbor FGFR amplifications, which is often an indicator of potential dependence. And when we looked at the growth of these different breast cancer cell lines, so MCF7 is an ER positive as a control, MCF10A is a basal-like immortalized breast line that's non-tumorigenic, and compare that to a number of triple negative breast cancer lines with different FGFR alterations. What we found was a range of sensitivity to this potent FGFR inhibitor. So cell lines like MCF7 are completely resistant, MDMA231 completely resistant, MCF10A is completely resistant. In contrast, this MFM223 that harbors an FGFR2 amplification is actually quite sensitive. And then two cell lines, Cal120 and HS578T, which have FGFR1 amplifications, have for intermediate sensitivity. So we wanted to study what's determining the relative sensitivity and resistance of these different cell lines. And to do that, again, we undertook genetic screens using genome-wide CRISPR libraries to ask what are the genes whose loss are potential sensitizers to FGFR inhibition. And when we did that, you can look at the the genes that are essential, for example, so if you just compare Cal120, MCF10A, MCF7, MDMB231, there are a set of common essentials. Many of these are pan-essentials. And then you can see that, again, here blue is more essential, that Cal120 has a set, there are a set of Cal120 specific essentials. So what are these essential genes? What we found was that in control conditions, Cal120 is very dependent on FGFR1, again, consistent with the idea that this is a potential driver oncogene in this cell type. In the presence of the FGFR inhibitor, FGFR1 is no longer essential. So that again confirms and supports the idea that this inhibitor's effect is mediated through its inhibition of FGFR1, since if you inhibit the FGFR1 with the drug, its loss no longer has an effect. But what becomes more essential are components of the mTORC1 signaling pathway. So while these are only either not or not essential under control conditions, they become increasingly essential under FGFR inhibitors. And when we look to see whether there was any evidence by using specific guides to mTOR1, what you can see is that the effect of the FGFR inhibitor is increased in Cal120 if we knock out mTOR1. So what's going on? So we also looked at the signaling downstream of FGFR inhibition in Cal120 and HS578T, both by Western blot and by phosphoproteomics. And when we did that, what we saw was, if we focus on phospho-S6, for example, you see that at 1 and 3 days of treatment, there's quite good inhibition, but by 6 days, this has rebounded. We see the same pattern actually for phospho-ERK1 and 2. And we look at the phosphoproteomics, so this is looking at control conditions on 1 day of FGFR inhibitor or 12 days of FGFR inhibitor. What you see is that, first of all, FRS2, the direct target of the FGFR receptor, it's inhibited continuously by the drug. So it's not as though the drug is no longer inhibiting FGFR, it still is. But these downstream pathways seem to be reactivated. So you see both evidence of reactivation of the mTOR pathway and also the MAP kinase pathway after 12 days of treatment. So that led us to the idea, well, that if we could block this reactivation through the mTOR pathway or the MAP kinase pathway, could we see that there's a synergy? And while we didn't see any synergy by inhibiting MAP kinase, when we inhibited mTOR with everolimus, we saw quite good synergy between either davitinib or infigratinib, 2 different FGFR inhibitors. So we're interested to dissect, well, what's happening? Why is mTOR reactivated? What's the mechanism? What we looked for was what changes after treatment with the FGFR inhibitor at both the protein and mRNA level. And what we saw was that following several days of treatment, upregulation of both amino acid transporters like SLC7A5 or SLC3A2, and genes that are targets of the YAP pathway, including CTGF, sort of the canonical YAPT target. If we looked at Go analysis, we see that transporters for amino acids are very strongly upregulated following FGFR inhibition. So how is that linked to mTOR? So work from David Sabatini and others have shown that amino acids can play, amino acid sensing can play an important role in mTORC1 activation. And to try to demonstrate that that's what's going on here, we looked at amino acid intracellular levels following FGFR inhibition by targeted metabolomics in collaboration with Alex Toker's lab. And when we did that, what you can see is that several amino acids, in this case glutamine, arginine, and leucine, are increased following FGFR inhibition. And if we ask, well, does that alter mTORC1 activity by looking at phospho-S6, what we can see is that, again, in complete media containing amino acids, after several days, phospho-S6 is back to baseline. If we remove amino acids from the media, now we see the phospho-S6 is low and not increased by FGFR inhibition. But if we replete glutamine, leucine, and arginine, we get a partial rescue of phospho-S6. So we tried to understand, well, what's regulating this change in amino acid transporter expression. And to do that, we performed H3K27 acetyl-ChIP-seq after different times of FGFR inhibition. And what we can see as we go forward in time, and sort of coincident with the gain of therapeutic resistance to the FGFR inhibition, we see a number of gained enhancers. And we ask, what are the transcription factors that are likely bind to these gained enhancers? Using a tool developed by Cliff Mayer and Shirley Liu's group in the Systrome toolkit, we could ask, okay, what are the, which cystromes from which transcription factors overlap most with these gained putative enhancers? And what we saw here were YAP, TAZ, and T. So elements of the YAP T transcription complex. So ChIP-seq data from publicly available ChIP-seq data on these factors overlaps quite strongly with these gained enhancers. So how is that happening? Why are these enhancers activated? And to do that, we looked at the chromatin accessibility by ATAC-seq, and then compared that to both the H3K27 acetyl-ChIP-seq and the YAP-ChIP-seq over time. And what we can see is that as you go forward at 4 hours, 48 hours, or 12 days, by 12 days, you see a very significant number of newly opened regions that by ATAC-seq. And those are characterized by having H3K27 acetyl and YAP ChIP-seq. And not surprisingly, in those gained putative enhancers, they harbor the TEAD DNA binding motif. And if you do sort of gene set enrichment analysis, you see that those up peaks are coincident with a YAP signature. So I think this figure is really, to me, has been the sort of most telling, and asks what's happening at these YAP-bound enhancers following treatment with FGFR inhibitors. So at baseline, there's this level of H3K27 acetyl, and then acutely at 4 hours, the activity of those enhancers goes down very markedly. But by 48 hours, and certainly by 12 days, that's completely recovered. So the H3K27 signal at this YAP-bound enhancers rebounds after 48 hours of treatment, and certainly by 12 days, it actually overshoots. Here are a couple of examples at these amino acid transporters. So you can see that, for example, at this site, it goes down at 4 hours. By 12 days, it's back up some. This one maybe even better. You see under control conditions, it has this intensity. That's down at 4 hours of treatment with the FGFR inhibitors, but it's going back up again at 48 hours, and is in fact even more prominent by 12 days. So can we use this knowledge to help us develop new therapeutic strategies? And to do that, we either grew Cal-120s or a PDX model harboring an FGFR alteration. So again, if you look at control conditions, these Cal-120 cells grow at this rate. They are modestly inhibited by this FGFR inhibitor, BGJ398, infigratinib. Knockdown of YAP has really no effect on their growth, but the combination of knockout of YAP and the FGFR inhibitor very significantly inhibits their growth. In this PDX model, we were able to study a YAP inhibitor CA3, and here we looked at the growth under vehicle conditions. Is this black line. If you treat with everolimus alone, there really is no effect. The CA3 alone has really no effect, but the combination, and then infigratinib, the FGFR inhibitor, has a modest effect, but addition of either everolimus or the CA3 inhibitor leads to complete tumor stasis. And to look at what's happening in terms of signaling, we can look at phospho-S6 in these treated tumors. So under vehicle condition, there's not much phospho-S6. Treatment with infigratinib actually increases quite substantially phospho-S6, again, consistent with the idea of mTORC1 activation downstream of chronic FGFR inhibition. That can be blocked by everolimus. Everolimus alone obviously doesn't activate it. Giving the YAP inhibitor alone doesn't activate it. But if you combine the FGFR inhibitor with everolimus, you can block this reactivation of S6. But quite importantly, blocking YAP does the same thing. So that really says that YAP is required for the reactivation of mTORC1 downstream of FGFR inhibition. And that leads to the second type of feedback loop, in which we see that FGFR, while amplified and important in the growth of this subset of triple-negative breast cancer lines, that its inhibition leads to reactivation of amino acid transport through the reactivation of YAP-dependent enhancers. And this increase in amino acids leads to upregulation of mTORC1 and blunting of the effects of the FGFR inhibition. I didn't show you the data, but we have evidence that SNF complexes are involved in this chromatin remodeling. So most importantly, I want to thank the people who did the work. The story on the estrogen receptor feedback loop involving CSK, and then the PAC2 synthetic lethality was led by Tengfei Xiao when he was in my lab. He collaborated quite closely with Wei Li, computational biologist in Chili Lu's group. And then the second story on the FGFR feedback loop has been really spearheaded by Yihao Li, who's a postdoc fellow in my lab, together with help from metabolomics from Alex Tober's lab, and the proteomics from Steve Gigi's lab. And for all of this, we've had a lot of help from our Center for Functional Cancer Genetics, who really has facilitated all the epigenomic profiling and data analysis. And I'd be happy to take any questions. Thanks.
Okay, thank you, Miles, for this great presentation. So looks like we don't have yet questions. So while we're waiting for some questions to come in, I have a few questions on my own. So one of them is, FGFR, obviously it's the ligand is very commonly comes from the stroma. So have you assessed whether, this resistance or, these mechanisms and these feedback loops are actually even more complicated in a real tumor where you have all these interactions with the microenvironment and also the cell-cell interactions? And how would you tackle that?
Yeah, so that's interesting. I mean, I think you could look for FGFR expression, in the stroma, potentially by single cell approaches. We don't, when we looked at whatever amount of FGFR ligands present in serum, whatever that level is, the FGFR inhibitor is able to continuously inhibit FGFR. So we look at FRS phosphorylation, it's continuously inhibited despite reactivation. So it doesn't require reactivation at the level of the receptor for this feedback. Now, there could be, of course, other mechanisms involving, very high levels of ligand present in the stroma. There is also evidence that there could be even autocrine loops in some tumors where, the FGF may be coming from the tumor cells themselves. So we do see in some proportion of tumors amplification of FGFs. But I think these inhibitors are quite potent and really able to inhibit very well at the level of the receptor. And that's why, I think that the feedback is coming in further downstream primarily.
Yeah, okay. Another question is, this SNF5 complex keeps coming up repeatedly in all sorts of resistance, not just endocrine resistance, and in fact, like ARID1A and B, and these are one of the most commonly mutated genes in particular treatment-resistant disease. Yeah. So does that mean that these nucleosome remodeling in general plays a role in switching the cells? Is this switching to a different state, or is just kind of loosening the states that the cancer cells can occupy? And how could we target that complex?
Well, there are, have been attempts to look for synthetic, lethal, so in cells that lose, redundant components like BRG and BRM, there have been attempts to target the ATP ACEs. Many companies, I don't know where any of those stand currently. I think your idea that, the mutations in switch SNF might play a role in sort of transcriptional heterogeneity, and that might allow more different transcriptional programs to be sampled under different selective pressures is a good one. That, potentially, that under the selective pressure of just growth, but then under drug selection, mutations in genes that constrain the transcriptional space that a tumor might occupy, would be an attractive potential mechanism of resistance, like your work on KDM5.
Yeah, okay. And then the last question I have, so we know that there are many feedback loops, and resistance due to feedback loops has been a recurring problem particularly for targeted therapies. Right. The question is why are we still continuing to try to use single agents in the clinic when we know that we need combinations, and how can we change this? Because, companies have been very hesitant to run combination trials, especially, until they don't have an agent approved for single agent use. Yeah. And how are we ever going to make progress then? We all talking about combination therapies, and at the same time, we trying to have single agents approved.
Well, so I think that, there are two types of combinations here. There are ones where the combination is there to try to obtain some type of pathway synergy because of, for example, these type of feedback loops. So that hitting a linear pathway twice can have an advantage. Then there's a second type of combination. It's really the ones that, Tom Fry, whose chair my chair is named in memory of, that Tom Fry's idea of combination therapy didn't have to do with synergy in a specific pathway or target, but had to do with non-cross resistant pathways. And I think ultimately for the cure of cancer, that may be, a even more important thing to come up with. That is, regimens that involve hitting pathways that are non-cross resistant. So, in most, in in complicated infectious diseases like tuberculosis or HIV, or in tumor types, solid tumors, or even leukemias that are cured with chemotherapy or targeted therapies. In general, it takes 3 drugs. You really need 3 non-cross resistant targets, so that the probability of a cell, if it's 1 out of 10 to the 6th has resistance, or 1 out of 10 to the 5th has a resistance mutation to one gene, to one target, it's going to be 10 to the 10th to 2, and 10 to the 10th is probably how many cells there could be, but it's 10 to the 15th to get 3, and that's just not going to happen, or happen much less commonly. So, I think the key is having non-cross resistant, highly effective targets. What I talked about today mostly about how to make a specific target. So in the case of estrogen receptor FGFR, it's really one target in its pathway, and trying to augment the effectiveness of hitting that pathway by hitting the pathway twice. And while I was not a big believer in that previously, I think it does can also play an important role to hit the same pathway more than once to get the most out of a single pathway. But to get cure, we really have to hit 3 different pathways.
Yeah. Okay, we got a couple of questions now. So one question is asking about, are there mutations in a SNP5 complex subunits in triple negative breast cancer? And there is data by actually multiple labs that the YAP activation plays a role in resistance in triple negative breast cancer as well.
Yeah. So this is in triple negative. I think there are more. I think there probably are more switch SNP subunit mutations than actually ER positive, but I'm sure there are some. I just don't know the numbers in triple negative. But
yeah, I think there are. Some of the ARIDs are mutated also.
Yeah, they're more commonly mutated in ER positive to be. Yeah. But there are. But the one problem is, of course, as we talked about, is that triple negative is not so. ER positive breast cancer, luminal breast cancers are much more homogeneous. They are. Have a very strong dependency on the same TFs and same factors, and they have mutations in PI3 kinase and cyclin D amplifications, but not much else in terms of mutation. Whereas in triple negative, it's a grab bag of again many different. They often have PTEN loss, P53 loss. Because of that, they have more other mutations as well.
Okay. Another question also related to this, upstream regulators of YAP and TEAD, such as SOX2. Did you have yours as the relevance of SOX2, or did it come up in any of the screens?
That's a good question. I don't recall.
No.
We found SOX2, but I think that's an interesting idea.
Okay. And then another question is, if in the first CRISPR experiment, did the essential genes become more essential in estrogen compared to vehicle?
I don't think so, because we control for the we have to always control for is the number of population doubling. So essential genes, of course, look more essential the more the cells grow. So you really have to be sure you get the same number of population doublings to see the essentiality. But
yeah, I mean, maybe some proliferation drivers could.
Yeah, common essentials. Some may have been. That's a good question. But I think I don't recall any that looked really much more essential in the estrogen versus deprived conditions. Yeah.
Okay. And then there was another question on the SNP5. Is this changes contribute to the plasticity of the cells, which kind of we alluded to, isoluminous switches?
Yeah, I don't think we saw evidence of switch to a luminal phenotype. Again, I don't remember all the gene expression. I think there may be actually some switch towards a more mesenchymal phenotype.
Yeah, I mean, it may not be the whole population, and it may not be like a complete switch, but
right. So we have actually, I didn't share, but we have some to look at this. We've been doing single cell analysis, and I just didn't include those data.
Yeah, no, because this is kind of it's never black and white. There is many in between. So, and then the last question is like, are these pathways driven or modulated by hormones? I don't know which pathways they're referring to. Maybe the epigenetic regulators, the SNP5 or FGFR itself.
Yeah, So, FGFRs do play a role. I didn't go into it, but they have been identified in ER-positive breast cancer as playing a role, particularly in endocrine resistance. In the triple negative, I don't know of anything that's regulating them, any hormones that are regulating the FGFRs or switch SNF. We need, they didn't come up in the gene sets that we would think, as regulators of them. But it's a good question.
Yeah. All right. And then the last one is, if the knockdown of CSK have an effect on the growth in vitro?
Knockdown of CSK has no effect on growth in vitro, in the presence of estrogen.
Okay.
Well, in the absence of estrogen, it increases growth, but in the presence of estrogen, it doesn't have an effect.
Okay, I think with that, thank you again for the great presentations, and thank you everyone for joining us. And again, we post the slides at the website, so you can go back and if you missed something, can look at that as well. Okay, thanks. Bye-bye.